Okay, so thanks for the introduction and hi to everyone in the room and online. I am going to give you guys an overview of what we have done on image analysis techniques that generalize, for example, beyond the data you have available for training your neural networks. And although I'm going to talk about or present two neuroimage related projects, the principles underlying this work are more general and apply broadly. So my plan for this talk is to have three parts. First, I'm going to introduce domain randomization. That is why neural networks depend on the training data and how synthesizing images well beyond the realistic range helps. And then in the second and third part, I'm going to demonstrate this with two projects that use the strategy and other techniques to achieve brain registration and brain segmentation that apply to your specific use case. We're all aware how AI has introduced algorithms with remarkable speed and accuracy. I'm sure that most, if not all of you, use some form of AI in your research. And we're also seeing an increasing uptake across medicine. In radiology, for example, by now, most of the major scanner manufacturers have introduced some form of AI algorithm into their reconstruction systems. However, compared to the more general field of computer vision, in our field, data sets tend to be of relatively narrow scope. There are a number of factors for this, such as a modality fragmentation. Just think of all of the different MRI sequences out there, or cohort stratifications, because typically what you cover in a data set is relatively narrow. So the list goes on, but my point here is that for these factors, models tend to generalize poorly. For example, if you train a neural network to skull strip a T1-weighted MRI scan, that network will not work well on PET scans. Now, you could, of course, include both of these data types in your training set, and this would work, but that model would still not generalize to new unseen types of images, like these clinical quality stacks of thick slices with a different contrast. So how can we get our models to generalize to image types unseen at training? Well, we suggest to synthesize arbitrary image features from anatomical label maps or segmentations, and then train our networks using a loss on the underlying labels, because that would be invariant to the image intensity. And that's domain randomization. So this strategy uses a procedural data engine that assumes you have a set of label maps for training, but no images. And usually you can find label maps online that you can download. So the way it works is, first, we move and deform an input label map. Second, we sample a grayscale image from it by assigning a different intensity to every structure in the label map. And third, we corrupt the image quality by applying a series of artifacts, including things like blurring, downsampling, and noise. Now, importantly, all of the steps of this data engine are randomized. So if we use the same label map as input once again, we're going to get a markedly different looking output image. I would like to emphasize that the goal here is not to synthesize images that are realistic or to simulate realistic images. Instead, we want to really expose our networks to variability to get them to generalize beyond the data that you may have for training. So we want to generate images far beyond the realistic range. How can we use this synthesis approach for model training? So we propose a general strategy that at each training iteration first synthesizes a grayscale image from a label map. Second, we pass that image to your neural network and have it make a prediction. And third, we compute the difference between the network output and a label map derived quantity of choice. What that will be depends on your task, and we'll see examples in the next slides. Now, minimizing that difference across many iterations is then what's going to train your neural network. Importantly, you only need the label maps for training once you train your model, you can just run it on images. And so that wraps up the first part. You now know what domain randomization is. And the second part, we're going to use it for image registration with SynthMorph. SynthMorph is a strategy for learning registration without real acquired images. And it is also a brain-specific registration method that is agnostic to the specific sequence or modality you acquired your image with. It adapts the warp regularity, and it is also aware of the anatomy you want to register. And those will become clear in just a bit. So in this part, I'm going to discuss how we use the synthesis approach, domain randomization, and some other techniques to train models that work on your data out of the box. The goal of image registration is, of course, to map the content or anatomy in a moving image to that in a fixed image by estimating a spatial transform between the two. This transform could be affine, which means that we apply the same shift, rotation, scaling, and shearing globally to all of the image content, or it could be a dense or deformable vector displacement field. Whichever one it is, once you have that transform, you can now use it to bring the anatomy from one image into the space of the other. For example, to carry out comparisons between subjects. Now, classical registration methods repeat an optimization for every new image pair. These methods deliver good results and are backed by decades of solid theory. However, they tend to be a little slow. Sometimes they can take up to the order of an hour per 3D image pair, and the optimization needs to be tailored to the specific task. So that's classical methods. In contrast, learning-based methods learn a function from a data set that maps an input image pair to an output transform. That function is usually parameterized using a neural network. And once you trained it, inference, by which I mean doing registration, is fast. However, these methods tend to break down on unseen types of data. For example, if we registered a T1 weighted to a T2 weighted MRI scan, those are slices extracted from 3D data, so we're doing 3D registration, then you would expect that the registered or moved image has, let's say, ventricles that more closely match those in the fixed image. But a network that hasn't seen this contrast pairing before might actually fail somewhat catastrophically. And so on the right, you can see real 3D outputs of registration networks. Unfortunately, the differences that can trip up these networks are often or can be way more subtle. Just like in this case where we registered two T1 weighted scans. And here you would, of course, expect the corpus callosum to better match that in the fixed image, but certainly not your hindbrain structures to be completely crushed if you look at the brainstem, for example. All right, so those were some failure cases. Now, how can we get our models to perform robustly across these different types of images? All right, we're suggesting to use domain randomization to synthesize widely, or like to say wildly variable images, and then train our networks to align the underlying anatomical labels. We do this using the data engine that we discussed, where we synthesize grayscale images from anatomical brain label maps. But do we really need label maps of brains? Because if we also synthesize if registration networks learn to match anatomy based on shapes, and if we also synthesize the underlying label maps, then we can effectively train registration networks without any real data. Are random shapes good enough to learn registration of real anatomy? So we test this hypothesis by training a network with the following setup. At each iteration, we sample a pair of shape label maps, and from them, the corresponding fixed and moving grayscale images. We pass these images to our registration network that predicts a dense warp field. And we use the warp field to move the moving labels, which gives us the warped label maps at the right. And so this setup is unsupervised. which means that we do not have a ground truth transform. Instead, what we do is optimize volumetric label overlap using a dice-based loss term. All our deformable registration networks use a UNet-based voxel morph architecture that predicts a diffeomorphism. And we train the same architecture using acquired MRI scans, T1 and T2 weighted scans, and also test classical baselines such as ANS and NIFTY-REG for comparison. So does this work? First, we test a shapes network that only samples images synthesized from noise distribution. And what you see on this slide is a taste of the types of images that network sees during training. Second, we train and test a brains model that samples contrast synthesized from anatomical brain label maps. So how do networks trained that way perform and compare to baselines on medical images of real anatomy? So we test this on a number of different data sets using dice overlap coefficients as a way of measuring registration accuracy. So higher numbers are better. On this slide, red is classical methods, green is learning-based methods trained with different losses, and then blue is our strategy. So for within or same contrast registration, we find that the shapes model matches the accuracy of the classical baselines, whereas the brains model, trained on brains, outperforms all of the methods we tested. And that is, although none of them ever sampled any real MRI data in training. Yeah, that was within contrast, the cross contrasts, we find that both of the models outperform all of the methods tested. Sorry.
You were first. I wanted to ask you, so like this on the sports vector is trained both on brains and on that random labels, or just on the brain?
Just on brains.
But let's say it makes sense to do like train it on both types of data to get a model that is first trained on these random labels and so on, and then take that and train it also on top on the brains. Scale up with the performance, or
so we haven't tried this. It might. I would assume that knowing something about the anatomy and the shapes in brains seems to help registration accuracy. So if you train on both of these types, it's possible that you get a network that applies to other anatomy, while it also does well on brains. So I mean, that's an interesting experiment. All right.
I just had a question about the legend. I'm confused. So I'm assuming SM shapes, SM brain is sent more. What's VM?
So VM is voxel morph, which is essentially the same neural network, but it's trained on T1 and T2 weighted images instead of these synthetic images.
What's the difference between TC and NMI?
Different losses used in training.
And then the other ones are just established registration methods.
Those are classical baselines, yes. All right, thanks. Okay, yes, and I think I was going to say that on the right side for cross contrast registration, the green learning-based methods that are trained on T1 and T2 weighted images, they were actually trained on same contrast pairs, so they haven't seen these contrast pairings in training, and that's why they break down. All right, so seeing these results, we wanted to understand how gradual changes in contrast affect registration accuracy for otherwise identical data. And so what we have here is registration pairs where, as you go towards the right, what we do is change or decrease the flip angle of the moving image, because those are contrast you get with a flash sequence, which I believe is gradient echo sequence. So the effect of this is that the moving image contrast evolves from T1 to proton density weighted on the right. And so at each flip angle or contrast pairing, we now run registration across a number of pairs and compute accuracy. So we find that our models in blue are most robust to this effect, while most of the baselines we test break down. So we wanted to see if this translates across pulse sequences. We just looked at FLASH, but it gets even more interesting for MPRH, a 3D sequence, where now we decrease the inversion time of the sequence as we go towards the right. And so here the effect is that different tissues or tissue classes are attenuated to a changing degree. So again, we measure accuracy at each inversion time, and we find that the models trained on synthetic data are almost invariant to these contrast changes, whereas the other methods decrease. Okay, so we get our networks to generalize beyond a specific type of real MRI contrast at training. But there are other problems that limit the utility of deep learning for a practically usable registration tool. For example, adapting the warp smoothness. Because deformable registration is generally ill-defined in that it is always a trade-off between similarity and regularity. Because you want your registered or moved image to most closely match the fixed image in appearance, but you also want your transform to be plausible. Without that plausibility, it's possible that your algorithm finds that there is a few voxels at the back of the brain that most closely match some structure at the front. You most likely don't want to have such a transform. So the way that virtually all of the registration methods out there address this, and that's deformable registration specifically, is that they optimize two objectives or two loss terms. There is a similarity term that encourages similarity in appearance between the images. And second, there is a regularization term that encourages smooth warps. Now, the relative weighting between those two terms is controlled by a hyperparameter, typically called lambda. And hyperparameter just means that it's set before training. Unfortunately, the optimal value of that parameter varies with many things, including the specific anatomy you're interested in, the type of like the population you have or type of data set, and also on the registration task. And that's on the example I have on the right, where you can clearly see different optimal lambda values for whether you do within subject or cross-subject registration for data from the same data set. Because it's a hyperparameter, changing that value of it typically requires retraining, which means that if you take a model that someone else trained on some data set and you test it on your data, that model will be suboptimally regularized and thus likely underperformed to what it could do with a proper lambda parameter. So we wondered, how can we make a model take lambda as an input? Because if we get our models to learn the effects of this hyperparameter, then we can efficiently choose the optimal value for your data at test time, so without retraining. So how can we get our models to learn the effect of lambda on registration? We suggest a training setup where, as before, we synthesize grayscale image inputs and pass them to our registration network. That network predicts the warp field that we use to move the moving labels. So until here, nothing has changed. In addition, we now also sample a lambda value from a uniform distribution and pass it to a second network that we're going to use to call hypernetwork. And what this network does is that it takes lambda and from it predicts the weights of the registration network.
Wait, so is it embedding them in latent space? Or just being fed in as another to the CNN as like another parameter?
So it's two separate networks in effect. We don't do anything in a latent space. We just have a network that takes in lambda and outputs the parameters of the registration network. So what that means is the registration network doesn't have any learnable parameters at all. They are an output of the hypernetwork.
Oh, okay.
So as a result, for a given lambda value, the hypernetwork outputs a specific registration network that is appropriately regularized for that lambda value. What does this look like in practice? So if we register this moving image on the left to the fixed image on the right here, then you can see for each lambda value what effect that parameter value has on the regularity of the displacement field. And similarly, what the effect of applying this to the moving image is. So at low lambda, we emphasize image similarity, which means the warp field is irregular and we have a strong effect on the image. If lambda is high on the right, then we emphasize regularity. So the field is smooth and we have less of an effect on the image. So in training, we just randomly sample from a uniform distribution. So can be whatever your 32-bit float numbers resolve. Right. Okay. And so we wondered, like given those qualitative results, how does network trained this way actually compare to baseline methods as before? So we tested this using a similar setup, again, with volumetric overlap as a proxy to registration accuracy, where higher is better. And so on this slide, blue is classical methods. Orange is a network that was separately trained by the authors, because we wanted to test off-the-shelf registration accuracy that you can get on your data, and green is our method. So we find that a model trained that way outperforms, is able to outperform all of these baselines across various different dataset and contrast combinations. And that is with a single mid-range lambda value that we did not optimize to the specific data sets, but you could. Okay, so we got our networks to adapt. Was there a question? No.
Yes.
Oh.
Quick one. So in your SynthMod, when you're basically, as you said, in the noise distribution or generator, basically when you're making all this synthetic data set, do you use the same number of training samples when you are training your SynthMod, or you use much more compared to the, say, the traditional or conventional deep learning methods, or do you use much more?
Yeah, we don't. We use much more. So all of the registration network results I'm showing come from a training set that includes label maps from 100 subjects, and we synthesize images on the fly. So every batch will essentially be different than any other batch.
Which makes sense. I mean, you're using synthetic data to have much more data than you have, but anyways, yeah.
Right. So I think going back to the very first plot where we had the same networks that were trained on real data or synthetic data, those also used 100 images. I mean, 100 different subjects that you can, of course, pair up any way possible. So you get like 10,000 combinations. But we also ran an experiment that I'm not showing today where we increased that to, I think, 5,000 subjects, and it didn't noticeably increase registration accuracy.
Because I was wondering if you just, as you were saying, just you take, I mean, sorry, this might be getting into too much, so we can put it later, but
sure.
If you just start swapping batches between, as you were, maybe you were alluding to that, between subjects, like with a more traditional, conventional way, in a way make it synthetic, but just from the data you have, not from a data generator or noise generator distribution, you were saying, will that also work? Basically making your algorithm more robust by changing the labels or batches between, well, in a reasonable way, I guess, between subjects, something, basically augmenting your data, a data augmentation technique.
So data augmentation generally helps. Question is, what do you do to the image intensities? Because in this synthesis approach, one of its major strengths is that you get networks that are agnostic to the contrast. But you can also augment the intensities in your real images, which I'm going to talk about in just a bit.
Oh,
Thanks for the question. Sure.
So how large the registration network is?
How large it is? So for these For these experiments, I found that I needed relatively large networks where just like UNets that had 256 filters on each level. And then I think I forget what I showed, probably 4 down sampling levels and 4 up sampling levels. So they seem to need larger capacity to be able to map the different intensities.
I'm curious about that. You to train to get the hyperparameter, the lambda.
Right.
So is it efficient compared to when you tune like the optimizer? Because in the optimizer, it already had a randomization curve, so we can tune it. So have you compare by your experiments with tuning the hyperparameter in the optimizer?
So I'm not exactly sure what you're referring to. The hyper network this is a different network. So that one is just kind of a simple MLP that had, I think, between 4 and 8 layers. I forget. That each had like 32 different units.
Yeah. I understand about the network, but you propose that you you develop a network to train to get the optimal lambda. So, and I'm curious that have you compared with the regularization curve in the optimizer?
So you're asking if we couldn't just learn the optimal parameter, I guess. We haven't compared this, but good point. The point of this setup is that you do not have a single fixed lambda, because if you learn the optimal lambda value, then it would be optimal for your training distribution. But if we now take this and apply it to Lila's data, and she might need a different lambda value. And this approach essentially learns kind of a continuum of networks where for whatever value you give it, you get the appropriately regularized output network. So you can adapt it after training.
So this, for example, this is lambda different for every subject. Once you train, so say you want to register, say a new data set for your 100 subjects. The data set is going to choose a different lambda for every of those 100 subjects, or is going to be one lambda for all these 100?
So this approach wraps the regularization problem in an SEP, so somebody else's problem, which means that so we have a default value of 0.5. If you don't specify anything in the setup, then it will just use a mid-range value. But what is optimal for your data, that will vary with every subject. So you could optimize that on each subject. In practice, what you'd probably do is, take some subset of your data set as like a validation set, and then optimize that. You could run gradient descent on it, and then you would just use an optimizer to just find whatever is appropriate for your data set, and then use that for the rest of the subjects.
But like the way it goes, you've got that the value plus is not that. Say the network picked a different.
No.
Once you applied it, when you tested it, so you said you tested it with a mid-range value.
Yeah, so there the value was fixed, but you could actually optimize it for every subject or every data set, and then you would get a higher accuracy, most likely. Thanks. Okay. All right, so that wraps up the bit about adapting warp regularity. Another fact that complicates use of registration algorithms in practice is a dependence on segmentation in preprocessing, because accurate brain registration generally hinges on skull stripping. Virtually all registration methods use a global similarity objective, image-based registration methods that just optimize similarity of appearance between the entire image content of the moving and the fixed image. But of course, if your brain moves independently of other structures in the image, such as the neck, then your brain-specific registration accuracy will not be optimal. And so that's why most registration algorithms expect you to skull strip the data that go in. Without skull stripping, if you ask methods to register images to this fixed example here, you may get slightly less accurate results, but you may also end up with catastrophic failures. And those examples are from both classical and learning-based methods, and it applies to affine and deformable registration alike. So we wondered, how can we make our registration networks aware of the anatomy you want to register? And it turns out that within the synthesis-driven framework, what we can do is synthesize input images for training from all of the labels in your label maps, but then optimize alignment of select anatomical structures only. Right, so in training, now we switch to affine registration. That means we could synthesize input training images from all of the labels in full head label maps. So clearly see there are facial structures and the neck in those images. And then we pass them to our network, which now predicts the global transform. And we apply this to the moving label map. But now what goes into the loss, we adapt a little bit, meaning we only retain brain labels. And because we're doing affine in this case, it's actually sufficient to remap to major tissue classes, gray matter, white matter, and cerebrospinal fluid. Affine registration also requires a different network architecture. And so briefly, what we use in this setup is an encoder that takes in a single image and then derives a set of learned feature maps from it that we reduce to centers of mass. So we get a set of points in 3D space. Separately, we process the fixed image, get feature maps, and also corresponding centers of mass. So we end up with two corresponding sets of points in 3D space, and we can just fit an affine transform that optimally aligns those points in a least squares sense. And there are closed form solutions for this. So let's look at some results. And we train a network that, as I said before, uses label maps from 100 subjects. Just to show the variability of this approach, this slide shows you images that were generated from one label map only. And so in the affine context, we apply rotations and shifts and so on in 3D. So the variability on a slice appears greater. All right, so how's the performance on medical images of real anatomy? We find that a network trained this way robustly aligns the brain in the conventionally school-stripped same contrast images, but also in full head images that were not school-stripped. Because of the synthesis approach, it generalizes to cross-contrast pairings, and we also find that it works on pediatric scans, including some that had motion artifacts or where the face was taken out for privacy reasons. So they were defaced, I guess you'd say. And it extends to clinical quality scans of thick slices with pathology. So, well, given those qualitative results, we then wanted to see what the effect of school stripping or the lack thereof is on baseline methods. So what we did is we ran a number of methods twice, once with school stripping as expected, and then a second time without that anatomical pre-processing. And we recorded the reduction in registration accuracy. So in this plot, lower is actually better. And we find that although a deformable registration suffered a little bit from it, it was more robust to changes in school stripping, whereas affine methods decreased by up to 10% in accuracy. And a network trained the way I just presented, where you used only brain labels in the loss, is actually largely invariant to school stripping. So you can run it on unprocessed inputs. All right, we put all of these technical developments and combine them into a registration tool that lets you register any brain MRI contrast right off the scanner. The tool is available at synthmorph.io. It does affine, rigid, and deformable registration on any sequence without pre-processing and retraining. And you can, if you want to, adjust the warp smoothness to your specific data. And I mean, for local users, this is also available in the local free software installs as MRI_synthmorph. Now, in terms of limitations, the tool expects that you generally have two mostly intact hemispheres a little bit of cropping is okay because that's also a part of the augmentation. And it has a focus on cross-subject registration, which doesn't mean you can't run it on longitudinal data, but specifically the affine and rigid parts might be a little less accurate than what you would get from a specific method tailored to that. We have a couple of resources online, including a demo that takes you through the steps of training a network on geometric shapes only. All right, so that wraps up the second part on registration. In the final and third part, I'm now going to present a project where we use domain randomization and targeted design choices to achieve infant brain segmentation with BabySeg. So BabySeg is a tool for segmentation in infants and young children that works without pre-processing, and it generalizes across input types and number of inputs that you might have available. It does segmentation of 21 structures, anatomic structures in 3D across the first 5 years of postnatal development from any number and combination of input, contrasts, and resolutions. All right, so in this part, I'm first going to touch on some of the challenges associated with neuroimaging in pediatric populations and why these make segmentation difficult. And then second, I'm going to present how we address them with BabySeg. By segmentation, I of course mean algorithms that take in 3D images like the one in the top and output a segmentation map, as I have in color here, that delineates various anatomical regions. We can then use those label maps to do morphological analyses, such as tracking brain development. We can compare this across populations, or often we also like to inform downstream algorithms, such as those that fit cortical surfaces. But cortical surface meshes, I guess. All right, so if segmentation goes wrong, anything can happen really. You might end up with a completely empty label map. Often you also might find structures in the background, like the red cortex or white matter on the left and in the center, or on the right, misplaced structures such as the orange cerebellum in the right ventricle, which doesn't belong there. And all of those examples are results from dedicated pediatric methods, deep learning methods. Okay, so why is neuroimaging so challenging in pediatric populations? One of the major challenges is, of course, motion, which can introduce artifacts if your subjects struggle to remain still for the duration of a scan. There are, of course, various mitigation or correction techniques. Simple ones would be swaddling babies and imaging them while they are asleep, but they are not always effective. And so the result is that your data can become unusable, or often you also may end up with sessions that do not include the full sets of contrasts, because many pediatric studies actually aim to acquire both T1 and T2 weighted scans. Of course, the size of children are generally smaller than adults, and so the field of view often includes non-brain anatomy beyond the neck, such as the torso and extremities. And speaking of the size, the major folding patterns of the brain are already in place at full-term birth, so around 40 weeks gestation, but the size still roughly doubles across the first 2 years of postnatal development alone. And during that time, the myelination of the white matter is in progress, which has profound consequences on the imaging contrast. So for T1 weighted MRI, for example, the contrast is such that the cortex is, at birth, it is bright, whereas the white matter is dark. Around a year later, the cortex is dark and the white matter is bright. And so consequently, the contrast has to invert at some point in between, which generally happens around 6 to 8 months of age. And during that time, you have very low contrast overall, and the contrast can also differ across the brain or a specific structure. Just like in this case at 4 months, where the cortex is bright at the front and it's dark at the back of the brain. Okay, so this is T1 weighted imaging. In T2 weighted MRI, we observe a similar but opposite effect, where the cortex is dark at birth. At year 1, it is bright, and so consequently, contrast inverts in between. And it's precisely for this variability in contrast that many studies aim to acquire both T1 and T2 weighted images. All right, so in sum, infant brains are not merely scaled down adult brains, and it easily takes an expert to about a week to annotate a three-dimensional image with the structures I'm showing here. Because of this, there is a real need for dedicated pediatric segmentation methods that are robust to all of these sources of variability, especially if you do not want to develop and run a different method for each combination of age and image contrast. So there are a number of pediatric methods out there. Unfortunately, many of them suffer from a combination of limitations, such as a focus on narrow age ranges or a focus on specific imaging contrast. And in addition, few of them actually focus or emphasize usability and runtimes. And I could spend a very long time on these because those were some pain points when I carried out baseline comparisons. I just go with one example, which is a method that required bits format as input, which is probably a generally useful format if you're sharing data sets publicly, but it means that to segment a single image, you need to understand that specification. You need to create a multi-level directory hierarchy. You need to place your image at the right point, and then you need to find the result elsewhere. All right, so that wraps up the imaging challenges. How do we address them with BabyCell? First, we wondered how we can train networks that are invariant to these, to the variability in soft tissue contrast. And of course, we're going to use domain randomization to synthesize arbitrary contrasts from label maps. That takes us back to the data engine I presented, where we synthesize variable images from label maps. And we extend it by adding random geometric shapes to the label map background to build robustness to non-brain structures in the image. However, for this specific project, we also wanted to maximally benefit from all of the real data we had available. And that goes back to a question we had earlier. So we extend this data engine to apply a similar series of steps to acquired scans, where first we sample an acquired MRI scan, and then we move and deform it randomly to get a new position and orientation and anatomy in the field of view. Next, we randomly remap the intensities to create a new artificial contrast. And third, we apply heavy augmentation techniques to arrive at the final output image. So how do we use all of this for training? Here the setup is slightly different. At each iteration, we sample a label map and all of the associated spatially registered inputs that we have from a session from one subject. We deform all of these inputs in the same way, adding random shapes to the label map. Then we pass these inputs to our, well, actually, I forgot this step. So we synthesize arbitrary contrast from all of these inputs, and then we pass these inputs to our segmentation network that predicts an output label map. Now, importantly, the number of inputs is variable in training, and the network outputs a single label map. This setup is supervised, which means that we now compare the labels to the ground truth label map. So why do we vary the number of inputs in training? And that goes back to the beginning where I said that many studies acquire multiple contrasts. If these are available, you would certainly like your segmentation method to exploit all of the information you have. So one way of doing this, one straightforward way, would be to pass those channels those inputs as channels to your CNN. And that would work. However, a network trained that way would be incompatible with any session that, for whatever reason, only has one of the contrasts. And similarly, if you have a repeat scan, good quality, you also can't include it. And so in this work, what we did was use a mechanism, a group convolution mechanism, that is flexible with regard to the number of inputs. So the goal here is to share information from and across a group of input images while keeping the size of that group flexible. The way we do this is we apply convolutions with shared weights separately to each of the input images. We derive a summary statistic. Here it's just the mean over input images that we also convolve. And then third, we mix that mean representation back with each of the inputs. That way, we can have interaction between all of the input channels, but the number of channels is not hard-coded. So the way we use this in our network is that we use a simple UNet architecture where we apply 2 of these group convolution layers at each of 6 downsampling levels and then 6 corresponding upsampling levels. That way, for n inputs, whatever n is, you get n outputs. And because we only want a single label map at the end of it, we use a group fusion layer that outputs a single one-hot encoded label map. All right, so we talked about the BabySec methods. Let me quickly show you some results, which takes us back to my favorite slide, showing the variability of the training data, which we generate from 1,000 subjects. In the interest of time, I'm not going to ask you to identify which of those images are synthetic and which are real. All right. So how does a model trained like this perform on real images? We find that BabySec accurately segments the brain and extremely preterm neonates. It also extends to newborns from different data sets with various quality levels, and it extends up to 5 years to subjects of up to 5 years of age, which is roughly the time when adult-focused baselines become reliable if you have good quality data. And importantly, we can run the same model irrespective of the number of input scans that you have, just like on those 2 rightmost examples where we do not have the T2 weighted image. So based on those qualitative results, we of course are curious how that model compares to baseline methods in a more quantitative way, excuse me. So we tested this using a data set that included more than 100 images from 4 cohorts of varying resolution, ages, and T1 and T2 weighted contrasts. Importantly, we also have a manual or semi-manual label map that for each image that allows us to compute registration, segmentation accuracy, excuse me. So we test a number of dedicated pediatric segmentation baselines, except for SynthSeg, which is a local method that most of you will know. They use similar synthesis techniques in training, but it has a focus on adults. All right, so let's look at some results here where we measure accuracy again as dice, so higher is better. And at the top, I'm denoting the methods where the subscript shows you what the inputs to each of those methods are. It's either T1 or T2 or both of them together. So we find that BabySec in pink generally ranks among the 2 best performing methods, but what those methods are varies by dataset and input combination. Importantly, we avoid gross inaccuracies of well below 70 dice points, with which virtually all of the other methods produce for at least 1 dataset. Now, it turns out that if you provide 2 images instead of just a single contrast, then generally that slightly boosts accuracy by about 1 or 2 dice points, except for this method called BibsNet, which we found struggles occasionally with the cross-modal registration it does internally, and we can't switch that off. All right, so given the robust performance, we were curious how segmentation accuracy evolves with age at scan, which we then tested using a subset of the DHCP dataset. Because for DHCP, we have relatively dense age sampling, meaning we have a scan of a neonate at each post-menstrual age. Higher, again, is better. And we find that as you move towards the left side on this slide, accuracy tends to decrease with younger age, whereas BabySec remains the most robust. And finally, we wanted to test this model on clinical quality images of artifacts contrasts and slice thicknesses that are atypical of the publicly available research-focused data sets out there. And we find that, as you see in the second row here, the map model predicts reasonable label maps for these previously unsegmented images. So just as we did with SynthMorph, we made BabySec into a hopefully easily usable tool, and let's just segment any infant brain MRI scan right of the scanner. You can find this method at babysec.io. We have a mechanism that is compatible with most of the major container platforms. It lets you segment infant brains across the first 5 years of postnatal development from any number of input images that can squeeze into your memory of any resolution, and that without pre-processing or retraining. The limitations are similar to those of SynthMorph. So here we expect the inputs to be nifty format images that are assumed spatially aligned. And currently we predict a single label for the left cerebellar hemisphere and one for the right only, and the same for the ventricles, but we're working on extending the label set. Okay, and so to wrap up, I would just like to use this opportunity to point out that we have yet another easy to use deep learning tool, SynthStrip, that lets you school strip full head brain scans acquired with a variety of different modalities. That's available at synthstrip.io. With this, I would like to thank all of the lovely people and funding sources that made those projects possible. And remember, deep learning methods have many advantages, but they are limited by assumptions made and the data you have available for training. Now we change this by removing training time assumptions through image synthesis and targeted design choices. We show that it's possible to get our models to generalize to image types unseen at training, to tune hyperparameters efficiently, and to adapt to any number of input images that you can squeeze in memory. So that means that you can now benefit from these advantages from deep learning, even without requiring high-end computational hardware or machine learning expertise. So I would like to thank you all for listening and encourage you to check out those tools if you haven't yet. Thank you.